etkinizi bana verin.
Structurally, most bacteria consist of a cell membrane surrounded by a cell wall, and for some bacteria, an additional outer layer. Internal to the cell membrane is the cytoplasm, which contains ribosomes, a nuclear region, and in some cases granules and/or vesicles. Depending on the bacterial species, a number of different external structures may be found, such as a capsule, flagella, and pili. In gram-negative bacteria, the gap between the cell membrane and the cell wall is known as the periplasmic space. Most gram-positive bacteria do not possess a periplasmic space, but have only periplasm, where metabolic digestion occurs and new cell peptidoglycan is attached. Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid, alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent the bacterium from bursting when fluid flows into the organism by osmosis. Synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria and thus have a much thicker cell wall. Beta-Laktam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin-binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics are therefore considered bactericidal agents. Bacterial resistance to beta-lactam antibiotics may be acquired by several routes. One of the most important mechanisms is through a process known as transformation. During transformation, chromosomal genes are transferred from one bacterium to another. When a bacterium containing a resistance gene dies, naked DNA is released into the surrounding environment. If a bacterium of sufficient similarity to the dead one is in the vicinity, it will be able to uptake the naked DNA containing the resistance gene. Once inside the bacterium, the resistance gene may be transferred from the naked DNA to the chromosome of the host bacteria by a process known as homologous transformation. Over time, the bacterium may acquire enough of these resistance genes to result in a remodeling of the segment of the host DNA. If this remodeled DNA segment codes for cross-linking enzymes, i.e. penicillin-binding proteins, the result is the production of altered penicillin-binding proteins. These altered penicillin-binding proteins can still cross-link the peptidoglycan layers of the cell wall, but have a reduced affinity for beta-lactam antibiotics, thus rendering the bacterium resistant to the effects of penicillin and other beta-lactam agents. This transfer process has resulted in penicillin-resistant S. pneumoniae through the acquisition of genes from other naturally occurring penicillin-resistant Streptococcus species. A second important mechanism by which bacteria become resistant to beta-lactam antibiotics is by the production of enzymes capable of inactivating or modifying the drug before it has a chance to exert its effect on the bacteria. Depending on the bacterial species, the gene coding for these enzymes may be found as part of the host DNA or on plasmids, which are small, self-replicating units of genetic material. Bacteria are capable of passing these resistance plasmids to each other by conjugation. When two bacteria come into close contact with each other, a small channel is created between them, which allows one of the bacteria to pass a copy of the resistance plasmid to the other. If the plasmid is transcribed and translated, the bacteria will begin to produce inactivating enzymes. These enzymes, capable of destroying beta-lactam antibiotics, are known as beta-lactamases. In gram-positive bacteria, the beta-lactamase enzyme is generally inducible, resulting in a large amount of enzyme being produced in the presence of the drug. In gram-negative bacteria, the beta-lactam enzymes are produced constitutively, i.e., even when the antibiotic is not present. Gram-positive bacteria release the beta-lactamase enzyme from the cell into the extracellular environment where it inactivates the drug before it enters the bacterial cell. In contrast, gram-negative bacteria retain the beta-lactamase enzyme within the periplasmic space, resulting in a more efficient mechanism than gram-positive bacteria. Ultimately, the destruction of the beta-lactam ring of the antibiotic renders it incapable of binding to the penicillin-binding protein, and thus the bacteria become resistant to that drug or class of drugs. The DNA is shown as a circular double strand within the bacterial cell. Like the DNA of all living organisms, it contains the unique genetic code for all of the proteins required for bacterial survival. These include the proteins required for reproduction, growth, repair, and regulation of metabolism. It also codes for the 3 kinds of RNA that are essential for carrying out protein synthesis. These are known as ribosomal RNA, rRNA, messenger RNA, mRNA, and transfer RNA, tRNA. In order for the bacteria to begin protein synthesis, the double-stranded DNA molecule must first unwind and separate in the region which codes for the specific protein that is to be made. Only one strand of the DNA serves as a template for this process known as transcription. Transcription results in the formation of messenger RNA, mRNA, which is a mirror copy of the DNA segment. Once the strand of mRNA is complete, it will detach from the DNA template and in turn become attached to ribosomes. Bacterial ribosomes are made of a small 30S and a large 50S subunit. After the 2 subunits join together around the strand of mRNA, synthesis of the polypeptide chain begins. This step involves the aligning of transfer RNA, tRNA molecules, and sequence along the mRNA. Each tRNA carries a unique amino acid determined by the sequence of the tRNA, which, when aligned along the mRNA and ribosome, join together to form the polypeptide chain. This step is known as translation. The ribosome will continue to add amino acids to the growing polypeptide chain until it reaches a point along the mRNA that signals it to stop. At this point, it releases the finished protein molecule. Macrolide antibiotics, such as erythromycin, act as inhibitors of protein synthesis by attaching to the 50S ribosomal subunit. As a result, they block the ability of the ribosome to synthesize the polypeptide chain. By inhibiting protein synthesis, macrolides are considered bacteriostatic antibiotics. However, at higher concentrations and with lower bacterial density or during rapid bacterial growth, macrolides may be bactericidal. Changes or modifications to the 50S ribosomal subunit, i.e., the target binding site for macrolide antibiotics, will confer resistance to macrolides and sometimes other classes of antibiotics. This type of resistance may be of a high level. This mechanism of resistance is mediated by the erythromycin ribosome methylation gene, which is found on plasmids, or transposons, i.e., small genetic elements which are capable of moving from one bacterium to another and integrating into the host chromosomal DNA. Copies of the gene are transported to other bacteria via plasmids or transposons through pili-like channels. The gene is incorporated into the new bacterial genome. During the process of protein synthesis, this bacterium will transcribe and translate the genetic code of the gene, resulting in the production of a protein enzyme capable of methylating the 50S ribosomal subunit at a specific position. This altered 50S subunit results in decreased binding affinity for macrolides and other antibiotics such as lincosamides and streptogrammin type B. This pattern of resistance is referred to as the MLS phenotype. Because the macrolide antibiotic is unable to bind to the 50S ribosomal subunit, it is unable to inhibit protein synthesis, and thus the bacteria itself is not harmed, continuing to produce polypeptide chains of amino acids. A second mechanism of bacterial resistance to macrolide antibiotics is mediated by efflux pumps. These efflux pumps are encoded by the MEFA gene, which is a transposable element. Because they confer resistance to only macrolides and not lincosamides or streptogrammin B, they are referred to as the M phenotype. The efflux pumps are energy dependent and for S. pneumoniae result in moderate levels of resistance. These pumps traverse the cell membrane of the bacteria and function to pump out the macrolide antibiotic after it has entered the bacterium. It should be noted that for other bacteria, such as Staphylococcus aureus for example, a different efflux system, which is plasmid-mediated and encoded by the MSRA gene, results in macrolide resistance as well as lincosamide and streptogrammin resistance in some cases. Despite the presence of these efflux pumps, macrolide antibiotics continue to enter the bacteria. However, once inside the cytoplasm of the bacteria, these efflux pumps actively remove the macrolide antibiotics before they have a chance to reach their target, the 50S ribosomal subunit, and bacterial protein synthesis is unaffected. In this animation, we demonstrate the biology of DNA replication, leading to bacterial cell division in a gram-positive bacterium, such as S. pneumoniae. The DNA is shown as a circular double strand within the bacterial cell. Like the DNA of all living organisms, it contains the unique genetic code for all of the proteins required for bacterial survival. Bacteria replicate by a process known as binary fission, whereby one bacterium separates into two new daughter cells. However, before this can occur, the bacterium must make an identical copy of its complete circular DNA. DNA replication requires that the two strands of DNA separate so that the genetic code of the bacterium can be read and a new complementary strand can be created for each of the original strands. To accomplish this, various enzymes, known as helicases, break the hydrogen bonds between the bases in the two DNA strands, unwind the strands from each other, and stabilize the exposed single strands, preventing them from joining back together. The points at which the two strands of DNA separate to allow replication of DNA are known as replication forks. The enzyme's DNA polymerase then moves along each strand of DNA behind each replication fork, synthesizing new DNA strands, in red, complementary to the original ones. As the replication forks move forward, positive superhelical twists in the DNA begin to accumulate ahead of them. In order for DNA replication to continue, these superhelical twists must be removed. The bacterial enzyme, DNA gyrase, which is also known as topoisomerase II, is responsible for removing the positive superhelical twists so that DNA replications can proceed. DNA gyrase is an essential bacterial enzyme composed of 2A and 2B subunits, which are products of the GYRA and GYRB genes. This enzyme has other important functions which affect the initiation of DNA replication and transcription of many genes. With the combined involvement of these enzymes, an entire duplicate copy of the bacterial genome is produced as the two replication forks move in opposite directions around the circular DNA genome. Eventually, as the two replication forks meet, two new complete chromosomes have been made, each consisting of one old and one new strand of DNA. This is referred to as semi-conservative replication. In order to allow the two new interlinked chromosomes to come apart, another bacterial enzyme is needed, which is known as topoisomerase IV. This enzyme is structurally related to DNA gyrase and is coded for by the PARC and PARE genes. Topoisomerase IV allows for the two new interlinked chromosomes to separate, so that they can be segregated into two new daughter bacterial cells. This animation will demonstrate two mechanisms of fluoroquinolone action. Fluoroquinolone antibiotics, shown here, are synthetic molecules that are bactericidal. The potency of these drugs is greatly improved by the addition of a fluorine molecule at position 6, and thus the term fluoroquinolones. Fluoroquinolones rapidly inhibit bacterial DNA synthesis, resulting in bacterial cell death. Although much is known about the molecular events that underlie the action of quinolone antibiotics, much remains to be clarified. Fluoroquinolones act by inhibiting the activity of both the DNA gyrase and the topoisomerase IV enzymes. For most gram-negative bacteria, DNA gyrase is the primary fluoroquinolone target. Fluoroquinolones have been shown to bind specifically to the complex of DNA gyrase and DNA rather than the DNA gyrase alone. As a result of this binding, quinolones appear to stabilize the enzyme DNA complexes, which in turn results in breaks in the DNA that are fatal to the bacterium. A second mechanism of fluoroquinolone action is shown here. With some exceptions, topoisomerase IV is the primary target of fluoroquinolone action in most gram-positive bacteria, such as Staphylococci and Streptococci, with DNA gyrase being a secondary target. The separation of two new interlinked daughter strands of circular DNA is disrupted. The final result on the bacteria, however, is the same. Bacterial replication is disrupted and the bacterium breaks apart. The relative potency of different fluoroquinolone antibiotics, and thus their spectrum of activity, is dependent in part on their affinity for either DNA gyrase or topoisomerase IV, or both. One of the most common mechanisms by which bacteria acquire resistance to fluoroquinolones is by spontaneously occurring mutations in chromosomal genes that alter the target enzymes, DNA gyrase and topoisomerase IV, or both. The frequency with which these spontaneous mutations occurs may be in the range of 10 to the power of -6. The effect of mutations on the activity of an individual fluoroquinolone will vary depending on the number of mutations, the location of the mutations, and which target enzyme is affected. If a mutation occurs, either in the GYRA or GYRB gene, that alters DNA gyrase and results in a reduced affinity of the fluoroquinolone antibiotic for this enzyme. The organism will become resistant. Similarly, a mutation may occur that alters topoisomerase 4, either in the PARC or PARE gene, and results in a reduced affinity of the fluoroquinolone antibiotic for this enzyme, and the bacterial organism will become resistant. It is important to note that for some fluoroquinolones that have similar affinity and potency against both target enzymes, mutations in both DNA gyrase and topoisomerase 4 will be needed for resistance to occur. In resistant organisms, semi-conservative replication continues.